Hello and welcome to Microbiology. This is going to be our first lecture from Chapter 1, and that is an introduction to microbes. So get comfortable and let's tuck in. We're going to start with a decently simple question, and that is, what is microbiology? Well, we can use that word and break it down. The word micro comes from the Greek word mikros, which just means small, which makes sense. We know what micro means. It means that it's little, right? Biology, we want to break that word down even further too. Bio meaning life, and ology meaning it's the study of. So when we put it all together, it means we are studying small life, which makes sense because microorganisms or microbes are going to include small organisms like bacteria, archaea, algae, protozoa, fungi, viruses, and helminths. Now, when we say a microbe is small, we literally are talking about really, really tiny ranges of measurements, such as 20 nanometers up to 100 micrometers. So those are those numbers, and then there are coordinating units at the bottom of this slide. I do want to give you an idea of how small that actually is, where a micrometer is going to be 1/1,000,000th of a meter, and then a nanometer is even smaller than that, where it's 1/1000,000th of a meter. So these are pretty darn small. And why would we study these organisms? Because they're both easy and hard to study. One of the ways that we can use microorganisms is to look at heredity. For example, E. coli, a very common bacteria that you have probably heard of and we'll talk about quite a few times through this course, can reproduce a new generation of cells within 20 minutes. So that rapid reproduction is very interesting to observe, especially in these small organisms. So speaking of particularly small, this is a nice picture of a pin tip or a needle tip with bacteria on that needle tip. So the first picture on the far left is going to be that more zoomed out image. And then as we zoom in to the middle image, we can see those clumps of bacterial cells. And then when we zoom in even further, it kind of looks like a Cheeto cheese puff, but that is bacteria, individual cells of that bacteria located on the tip of that pin. Microorganisms can come in a few different flavors as far as how their cells are arranged. You can have unicellular, which means one cell, and each cell is going to be independent. You can also have multicellular, where the cells are undifferentiated, meaning that the cells themselves are independent as well, but there are many cells clumped together. A good example of this would be like algae. You can also have multicellular microorganisms, and mainly when we talk about this multicellular with differentiated cells, we're talking about kind of higher forms of life, maybe like an animal or a plant, something that has specialized tissues. And I want you to think of on yourself those specialized tissues. You have specific tissues for your heart, cardiac tissue. You have specific tissues for your lungs, your kidneys, your liver, skin cells, cells that make up your eyes. These are all different cells that have different jobs. They're differentiated, but they work together. And another important feature of these differentiated cells is that if you remove a clump of those cells and remove it from the organism, those cells are not going to be able to survive independently. because they can't survive independently. As opposed to if you took algae and you removed some pieces of algae or some cells of the algae from that main clump, they'll be fine. We're going to give you this idea of a blender test, which can kind of sound a little bit violent or traumatic, but bear with me while we talk about it. The blender test is if you take multicellular organisms that have undifferentiated cells and you put them in a blender, blend it up, those cells will be fine because, again, even though they came from a multicellular organism, they are independent of life by themselves. If you take a multicellular organism, like, I don't know, a bonsai tree, or a dolphin, or a regular snail, something like that, and you put that whole organism into a blender and turn it on, those cells are not going to be able to regenerate and they're not going to be able to function independently anymore because you have now taken all of those cells, those differentiated cells, and blended them up in a pattern that is not okay. As you can probably imagine, microbes are incredibly important to our planet and in fact have shaped much of the Earth's habitats and also assisted in the evolution of other life forms on the planet. A good example is algae and cyanobacteria, very early forms of them, produced oxygen as a byproduct of photosynthesis. This happened in such excess that it has caused what we now currently have as the Earth's atmosphere. So that is how much microbes have impacted this planet by changing our entire atmosphere entirely. So let's switch gears to a little bit of history as far as microbes go. Where did they come from and how do we know? So first, I want to set the scene for you. Gentle reminder that the Earth is roughly 4 billion years old, and a small skip and a jump from that conception of the planet about 3.8 billion years ago, we see from the fossil record that there is some form of a very, very early looking single-celled organism. From that single-celled organism, we end up seeing something that gives rise to a more familiar type of cell to us, which is called a prokaryotic cell. This is going to be cells that end up giving rise to bacteria and archaea. This would have been around 3.5 billion years ago. These first couple cells of prokaryotic cells that we see from the fossil record, we call the last common ancestor, or LCA. These cells would then eventually become what we've come to know and love only 2 billion years ago, eukaryotic cells. These appeared from that LCA and would eventually give rise to organisms like protozoa, algae, helminths, fungi, plants, and animals like us. Now, you'll notice that there's something that maybe you think should be on here that is not, and that is viruses. They are not included in this evolution, but we will talk about viruses soon. So hold on for that. It is coming very soon. Now, I said that there were two types of cells that were really important. We have prokaryotic cells and eukaryotic cells. What prokaryotic means is pre-nucleus. There is a very important feature about prokaryotic cells, and it is that they do not have a nucleus, nor do they have any of the classic organelles that a eukaryotic cell contains. They are generally much smaller than eukaryotic cells, and they are almost always single-celled organisms, but not always. Eukaryotic cells, on the other hand, are known as having a true nucleus. They contain a nucleus. They also contain a ton of other organelles that you've probably heard of in plenty of other classes, but we are going to gently go over that, like the Golgi apparatus, mitochondria, chloroplast, smooth ER, rough ER. We can kind of go on. There's a lot of different organelles. They're also generally larger, and the reason is they have to have the room for all of those various organelles. So they have to have a little bit of extra space for what they're packing. These can be single-celled organisms, but are often multicellular organisms that we have come to know and love. This is a gorgeous illustration of a prokaryotic cell with a bunch of various features, really nicely defined, which, hold on to this image. It's a good one to refer back to. We will talk about quite a few of these structures on the prokaryotic cell, but not for just a little while yet. Probably not in this chapter, but it is coming up. I do want to explain on this eukaryotic cell that what we're seeing is kind of a chimera of two cells being smooshed together in order to give you a very oddly comprehensive illustration. So on the left, we're seeing a lot of features of a plant cell versus on the right, it is all of the structures that you would see on primarily an animal cell. Plus a flagella. So we're kind of seeing a weird image here, but these are all of those structures laid out, and it's kind of an interesting image to look at when it comes to the different organelles and structures of a eukaryotic cell. As promised, I told you we would talk about viruses very soon, and here it is. So we need to make sure that you guys understand that prokaryotes and eukaryotes do not contain viruses. Okay? Viruses are their own individual thing. They are not prokaryotic or eukaryotic cells. Viruses are known as acellular. So any word that starts with a or an, typically means no, none, or not. So this is not a cell. I want to make that very clear. Viruses are not types of cells. They're smaller than any type of cell that we have currently. They are less than 450 nanometers. And remember, a nanometer is that smallest size, 1/1000th of a meter. These are going to be organisms that must have a host cell in order to live, essentially. But it is debatable if viruses are actually alive. Although we do comment that we can kill a virus or a virus is dead. So if we use those terms, is it technically alive? It's kind of up to you. One of the ways that you can think of a virus, because it's not quite a living thing, is that it's more like a machine than it is a living thing. What's odd is it's a machine that must have a host in order to reproduce, and that's kind of an odd feature of a virus itself. It's kind of parasitic in that sense. I mentioned before that prokaryotic and eukaryotic cells that we have today evolved from this initial cell called the last common ancestor, or the LCA. This cell was neither prokaryotic nor eukaryotic, but it gave rise to both types of cells, and it would be good for you to know when that did happen. These two types of cells, prokaryotic and eukaryotic, eventually would become the three domains of the tree of life. And the tree of life is going to be like a gigantic family tree for all living organisms on Earth, which I'm going to show you next. Make sure you know those three domains. Those are at the bottom of the screen. We have bacteria, archaea, and eukarya. Remember that bacteria and archaea are going to be prokaryotic cells, and eukarya are going to be eukaryotic cells. Now, I do not expect you to remember all of the organisms on this tree, but I do want you to remember those 3 domains, and this is just a nice way to kind of visualize the way that this tree is going to be formatted. We have bacteria on the left, we have archaea in the middle, and eukarya to the right. Where our various parts of our tree, our branches, are meeting, that is going to be a common ancestor. Now, where they all meet at the very bottom, that's going to be that main branch, and that is going to be where our last common ancestor would exist. The cell, that precursor to these cells, would have existed. So, something to kind of point out, when it comes to organisms that fall in the eukarya domain, we've had substantially more time to collect information and data about these organisms because we've been able to see them for much longer than we have bacteria and archaea. We just haven't had as long to study them because we haven't had the ability to, so the technology to be able to study those organisms. Now, that last tree was kind of the basic model of our tree of life, but it's actually substantially more complicated than what I showed you on that last slide. This is a much more comprehensive, inclusive, and complex tree of life that includes everything. Many more encompassing trees of life are going to be kind of interactive in a way where you can zoom in really far. But these guys are very complicated. I do not expect you to know it. I'm only showing it to you for fun. I do want to dive a little bit into the history of eukaryotic cells and how they came to be. Specifically, how do we have these cells that have organelles inside of them? So that leads us to the theory of endosymbiosis. And before we get started, I do want to give you a definition of what a theory is. A theory is a well-supported but very broad hypothesis, which is going to be very well supported as no one is able to reject that theory with something else to explain that phenomenon. So in order to explain the theory of endosymbiosis, I want you to imagine that many, many years ago, when we had that very early, something like a eukaryote, that last common ancestor type cell, it would have been potentially a cell that would have been consuming, engulfing, or you can think of eating other cells until it found potentially cells that were useful to it. Now, these cells would have been like little batteries so that it wouldn't have to consume as much in order to have energy. So these other types of cells may have been something like early mitochondria and early chloroplast. And what's important about these little guys, these organelles, is that they have everything to do with the production of energy. So imagine consuming a little battery and being able to rely on that guy and have a symbiotic relationship with that organelle. So it essentially just provides energy for you, and you provide safety for it. So it's a symbiotic relationship, right? Both parties are having a good time, essentially. So, how do we support this theory? Why do we think that this happened? Well, some supporting evidence when we look inside eukaryotic cells, we can see that the DNA inside the nucleus is going to be linear. Now, inside of prokaryotic cells, the DNA is going to be circular, and we will talk about this more eventually, but just so you know, that's kind of the basic there. So, eukaryotic DNA is linear chromosomes, And prokaryotic DNA is going to be circular. Okay? When we look at the DNA of mitochondria, it's kind of interesting to see, but it has its own individual DNA by itself. On top of that, the DNA that mitochondria contains that is different from the DNA inside of the nucleus, it's also circular. So this supports this idea that eukaryotic cells consumed other prokaryotic cells and then created this symbiotic relationship with those cells. So that is the theory of endosymbiosis. Kind of neat. You do not need to memorize the steps from this slide. It's just a nice illustration showing a very early cell and potentially the first endosymbiotic event occurring where this early prokaryotic cell, or more of like you can think of it as an ancestral eukaryote, is going to be consuming other types of prokaryotic cells and then collecting them as organelles. Now, I know what you're thinking. Great. That's fantastic, super interesting how organelles made their way into eukaryotic cells. But how did those eukaryotic cells become multicellular organisms? Well, initially, those eukaryotes with their organelles were probably single-celled and independent until they started to clump together and form something like a colony. Those colonies would then start designating certain cells to have special roles within the colony, and thus those cells became specialized, until eventually more complex organisms started to evolve, and the individual cells within those organisms would lose the ability to survive on their own and become differentiated, and thus something like us with differential cells like lungs, kidneys, cardiac cells, etc., and so on. This slide is not something that you need to memorize. More than anything, it's just kind of interesting seeing the timeline of events to get us to where we currently are on the planet. So 4 billion years ago is when the conception of our Earth started. About 2 billion years ago, about halfway point in our timeline, was when those first eukaryotic cells would have started coming around and enveloping those prokaryotic cells. And from there, when it comes to humans, we are pretty far down on that latter part of the timeline. The first human ancestors arose somewhere around 4 million years ago. That is just a blip in the timeline, let alone when modern humans would have started roaming the planet. So kind of interesting to see on a grand scale, makes you feel pretty small in the grand scheme of it all. Microbes, and especially bacteria, are a term that I'm going to give you, which is ubiquitous. This means that they can and do live everywhere on the planet. They live within the Earth's crust, the polar ice caps, on ocean floors, inside the bodies of plants, and especially inside our bodies, as well as outside of our bodies as well. They're going to occur in large numbers, which makes sense. Microbes make up a vast majority of life on our planet, and they can also thrive in areas that other organisms can't. And we'll talk about some of these conditions where you have extreme environmental conditions, and the only thing that does survive are these really interesting types of microbes. Microbes are going to have a lot of different byproducts as well as different uses that we have found for them. So historically, humans have always used microbes, or at least loosely somewhat understood that there are uses for microbes. Good examples include bread production from yeast, alcohol production through fermentation, cheese production, treating wounds aseptically, mining precious metals, sewage and water treatment, which we are going to have a lab that helps to show us that process, which is kind of interesting. And then more recently, we have been able to use microbes to help clean up man-made contamination sites, as well as oil spills. We'll talk about that here in just a bit. Now, aside from historical uses like making various types of foods and treating wounds, these are also ways that we currently use microbes. So genetic engineering, potentially you have heard of restriction enzymes, and if you haven't, we are going to talk about them primarily in the upcoming chapters. But we have stolen, and I use stolen in kind of a loose term, kind of more so we've borrowed the restriction enzymes from bacteria that is used to chop up DNA like specialized scissors, and then we use that to genetically engineer or modify organisms. We chop up the DNA using those specialized scissors that we stole from the bacteria. Kind of interesting. Another way is recombinant DNA technology. Kind of a silly example, but one I'm sure you can think of is like the glowing fish at the pet store. Those guys don't normally glow under a black light, not in the wild at least. So what we have done is transfer that genetic material from jellyfish DNA to those fish that are sold in the pet store in order to make them have a specific phenotype, that physical display that makes them glow. So that is recombinant DNA technology, and we've stolen that kind of from bacteria as well. And then we have bioremediation. A good example of this, I told you we'd talk about it again, is oil spills. So there are 3 species of microbes that eat oil, and the byproduct of those microbes is oxygen and water. So as they consume the oil and clean up a spill in a natural area, they're also producing things that we want in those natural areas like oxygen and water. So that does help us in the long run. Now, using the uses of microbes, we do have fields of microbiology currently in the planet. So the first field is going to be medical microbiology. So this is going to be our relationship, our personal relationship with microbes. So this could be anything from studying all of the microbes that we contain in and on our body. So the Human Microbiome Project is something we'll talk about a few times during this course, and how interesting that is, and how we're able to look at collective pieces of data on various people from around the planet to analyze what microbes are present where and in kind of what quantity. It's very interesting, as well as developing a means of disease prevention like vaccines and antimicrobials, maybe antivirals, better antivirals, or antibacterials in general in treating various diseases. So another field is environmental microbiology. So this is going to be the effects on the earth and atmosphere based on microbes. And Some stuff that we can do with these microbes is remove toxins from underground wells, chemical spills, toxic waste sites, oil spills, like we mentioned before. Bacterial enzymes can also be used as drain cleaners. Bacillus enzymes are used in household detergents to remove spots from clothing. Fun fact. And sewage treatment plants remove undesired materials and harmful microbes using various physical and chemical processes with the action of beneficial microbes that will eventually convert to carbon dioxide, nitrates, phosphates, sulfates, ammonia, water, and methane. Two more fields of microbiology used today include industrial microbiology. This is going to include the exploitation of microbes to create commercial products. So things like foods, vitamins, antibiotics, alcohol, acetone. A big organism within this group is Saccharomyces cerevisiae. That's going to be baker's yeast, a very common yeast and microbe used in winemaking, various other alcohols, as well as bread. Kind of interesting. Another field is going to be agricultural microbiology. This is going to be, and if you want to kind of think of it this way, kind of like GMOs, where it's going to affect livestock and plants. There is a type of corn called BT corn, and it uses the bacteria Bacillus thuringiensis, which has a property that is anti-microbial, as well as has a pest deterrent feature of it. And so they have modified the corn to contain these properties from the genes of the Bacillus thuringiensis, thus making the corn BT corn. This enables the corn to have a natural pest resistance as well as microbe resistance, which is kind of interesting that it has these properties. Thus far, we haven't found any links to BT corn having an adverse effect on humans, but it makes it to where you don't have to use a pesticide or an antimicrobial on the corn itself. There are 5 specialized fields of microbiology. We will talk about all of them in some detail. Starting with bacteriology, this is the study of bacteria. Mycology, which is the study of fungi. Parasitology, which is the study of protozoans and parasitic worms. Immunology, which is the study of our immunity. And virology, which is the study of viruses. As I said, we will talk about the specialized fields a little bit more in depth here in a minute, as well as throughout the semester. Now, it should come as no surprise that microbes are able to harm humans. When we talk about microbes that can cause diseases, we call this a pathogen. Now, there are 2 organizations that help to fight and study infections, and you need to know both. That is the World Health Organization, or WHO, and Center for Disease Control and Prevention, which is the CDC. Now, what is important to note is that infectious diseases are the most common cause of death in the US and worldwide. So both of these organizations are imperative in fighting those diseases and finding ways to better fight diseases, as well as to get out information to help in prevention or medicine. To emphasize how important the study of microbes are, there are a handful of diseases that were previously thought to be non-infectious, have now been found to have microbial origins. such as gastric ulcers with Helicobacter pylori. That is the type of bacteria found every single time there is a stomach ulcer. So it is always present. So there may be a relationship there. These are well-supported relationship examples. Diabetes also has a microbe. Schizophrenia has more recently been linked to the Borna agent, which is kind of like a virus. So I do want you to know a few of these examples. Make sure you are aware that some microbes that we previously, or conditions we did not think previously had a microbe associated with it, actually are starting to come out, that we are seeing a link from these diseases to certain microbes. In current times, we have been seeing emerging and unfortunately re-emerging infectious diseases. So new diseases like coronavirus, AIDS, hepatitis C, viral encephalitis, Ebola, and the Zika virus have a very severe morbidity and mortality rate, while older diseases like TB or tuberculosis, as well as measles and whooping cough, have started becoming a problem for various reasons. Tuberculosis has started to become antibiotic resistant, which is a very not good time, while measles and whooping cough have reemerged due to a fear of vaccines and a lack of vaccines being taken in various areas of the world. So when we see that herd immunity start to decrease, we do start to see those reemerging infectious diseases start to pop back up in certain areas. All right, we are going to start the 7 subgroups of microorganisms. What I need you to know for each one of these 7 subgroups is 1, the type of cell, and if it causes disease. So make sure you take notes on both of those. Naturally, our first is going to be the bacteria. This is a prokaryotic cell, so that is the cell type. It's mostly unicellular and also very small. Remember, it's going to be in micrometers, okay? But remember, our smallest is going to be in nanometers, so this is not yet the smallest of our organism types. They are everywhere in all environments. They're inside and outside of our bodies. They're used in food, industrial production, which we mentioned previously, and they do cause several diseases. There are about 5 million trillion bacteria living on the planet. And on the next slide, we're going to talk about an example I want you to know. You do not need to know all of the information about E. coli, but I do want you to know that E. coli is an example of bacteria. A couple interesting things about it is that in our bodies, it's located in the large intestine. It does serve a pretty important purpose of helping to break down food as well as produce vitamin B and K, and it protects us from harmful bacteria. And as long as it stays in our large intestine, then it's a pretty safe bacteria. It's kind of a bacteria that is our home team, if you want to think of it that way. Now, there are strains of bacteria, at least one that we know of, that can make us super sick. The other thing to make a note of is if E. coli travels for some reason from the large intestine to a different orifice or a different part of your body, it can cause an infection. So just because it's safe in one area, if it's put in a new area where it would not have previously been, that can cause a problem. Our next organism is going to be archaea. Archaea are going to resemble bacteria and have a prokaryote like cell structure. They can live in hostile environments that are particularly hot or acidic or possibly salty. We call these organisms extremophiles, depending on where they are. You can also have methanogens, halophiles, extreme thermophiles. It just depends on what we're talking about, but we will talk about some of those terms later too. Now, it is important to note that archaea are not associated with diseases as of now. Who knows, maybe that information will change eventually, but as of right now, we have not found any archaea that are associated with any known diseases. An example that I just want you to know the name, you don't have to know all the different things, is Methanopyrus. This is going to be an archaean that is located in hot water, such as the hot springs in Yellowstone. So these guys are going to create methane by combining carbon dioxide with hydrogen from rocks in geothermal springs and other hot springs, and they can have a tendency to give hot springs kind of a sulfurish, odoriferous smell. Next up is algae. This is going to be a microorganism that has a eukaryotic cell structure, and they're plant-like because they use the process of photosynthesis, turning light into energy. They also produce oxygen and carbohydrates, but it's important to note that even though they have plant-like qualities, they are not plants. So, very important thing to make a note of. They can be microscopic or macroscopic, meaning that you can see them with the naked eye. They can also be unicellular or multicellular, which is kind of a unique feature of a microorganism. They can be abundant in all kinds of water, soil, as well as live on top of plants. And it's important to note that some can cause diseases as well. An example of an algal cell is Diplonius. These guys can live in salt water, and what's kind of interesting about them is that when times get tough, they can go from being a single-celled organism to a multicellular organism that can help the other cells survive during those harsher times in their environment. Also, kind of an interesting thing is they are commonly found in exfoliating face and body washes. So you have probably, if you've used an exfoliating scrub, used little Diplonii, on your skin to exfoliate. The next microorganism is one of my favorites, and that is fungi. These are going to be eukaryotic cells that are going to use organic chemicals for energy. So they are not plant-like, they are not photosynthetic at all. So some interesting things about them is included within fungi, we have yeast, which are going to be an example of unicellular fungi, while molds and mushrooms are going to be multicellular examples of fungi. What's kind of interesting is that some of these microorganisms within fungi can cause diseases. Now, I mentioned this guy before. This is Saccharomyces cerevisiae, or baker's yeast. It is used to make bread. It can also be used in making alcoholic beverages, and that is because this organism is going to convert sugar into alcohol, so that fermentation process. The next microorganism is going to be protozoan or protist. So these guys have the name first animals. They are a eukaryotic cell structure. They are all microscopic and unicellular. Some examples include things you've probably heard of like amoebas, maybe some things you haven't heard of like flagellates, ciliates, and sporozoa. Some of these are known to cause several diseases. The example I have for a protozoan is the Vorticella. This is a really interesting protozoan that live in fresh or salt water. They can also live in moist soil, but they have an interesting hunting technique where as they use that bell you can see on the picture to sweep in and kind of like a mouth grab food, it can also use the stalk as a spring to bounce up and grab food. Another example of a protozoan that you've probably heard of is malaria, which is that organism that can be transmitted through the spit of mosquitoes and cause someone to be anemic due to a loss of red blood cells. Helminths are our next microorganism. They are eukaryotic cell types. These are going to be multicellular parasitic flatworms as well as roundworms. So if you can get through this slide without feeling a little squeamish, congratulations. You are stronger than I am. These guys are going to have multiple stages in life, which can also make them very difficult to treat. Because they can come in eggs or larval or adult stages that can be difficult to determine how to best get rid of helminths if you have an infection, because these guys do cause diseases. The example I have for a helminth is a tapeworm or cystota. These guys are very unattractive to look at, if you can imagine with that little picture there. What's interesting about these little fellas is that they are often found in digestive tracts in humans as well as dogs. And kind of a not so interesting and kind of more of an icky sort of piece of information I have about tapeworms is that in 2013 there was a diet pill that contained tapeworm eggs. And you might be asking, why would you ever? Well, if you have a little parasite in your intestines, that will help you lose weight. Maybe not in the way that you would want, because not only do you have a little parasite in you, that little parasite is reproducing like crazy. And in order to get rid of your little weight loss friends, you are going to have to defecate yourself pretty violently, or potentially, if it's extreme enough, we'll have to have surgery to remove them. Last but not least, in our subgroups of microorganisms is viruses. Now, gentle reminder that viruses are neither prokaryotic or eukaryotic. They are acellular. And these guys are going to be the smallest of all of our microorganisms, being somewhere around 20 to 100 nanometers. Remember, that's a very small measurement. These guys can have extremely simple structures, which include a nucleic acid and capsid. They are also known as obligate intracellular, meaning that they must live within a host, kind of like a parasite. And of course, as we probably are aware, viruses do cause several diseases, which we will talk about some of those coming up.